In this video, we're going to discuss human immunodeficiency virus, or HIV. This particular virus is the cause of acquired immunodeficiency syndrome, or AIDS. We'll talk a bit about the structure of the virus, its replication or biosynthesis, and then we'll finish our discussion talking about some of the antiretroviral therapies that have been used to treat HIV infection. The viral particle for HIV is enveloped, and it contains a cone-shaped capsid inside a spherical structure of matrix proteins. Within the cone-shaped capsid, we find 2 copies of single-stranded RNA associated with some proteins, including reverse transcriptase. On the envelope, we find 2 proteins, GP120 and GP41, which make up the spike, and these are going to be involved in attachment and entry of the viral particle to host cells. The reverse transcriptase is an RNA-dependent DNA polymerase. Let's take a look then at the HIV life cycle. It should be noted here that HIV primarily infects T helper cells. We'll come back to this point a little bit later on. But the infection begins when the spike protein on the viral particles binds to CD4 and to a co-receptor known as CCR5 or CXCR4. After this attachment step, we find fusion when the spike protein changes shape. The viral envelope and the host membrane will fuse together, which will lead to the release of the capsid into the cytoplasm. At this point then, the viral continues to uncoat, and that RNA-dependent DNA polymerase reverse transcriptase will convert both of the single-stranded RNA genomes into DNA. That DNA then will be transported into the nucleus, where it will then be integrated into the host genome. And this is performed by a viral enzyme known as integrase. At this point, the biosynthesis of the virus, its replication, its transcription, and its translation is completely regulated by the host. When viral particles are formed, what we find is that the virus will assemble at the cell membrane, and at this point it will bud to exit the cell, and it completes maturation after exit. In particular, HIV codes for a protease that does the final protein cuts or cleavages of protein after the virus has exited. At this point, after protease has cleaved all the viral proteins into their mature shape, the viral particle is now capable of infecting more cells. Let's take a brief look at the attachment and penetration of HIV into its host cells, the T cells. In this example here, we can find the spike protein in green and red. The green represents part of the spike known as GP41 or glycoprotein 41, and the red represents the GP120. GP120 interacts directly by binding to CD4, which is found on the surface of T cells. GP41 will interact with one of 2 proteins, CCR5 or CXCR4. Upon binding of the spike protein to CD4 and the co-receptor, what we will find is that the viral particle will begin penetration. In penetration, the viral particle is brought close to the membrane of the host cell, and then we find a fusion event in which the 2 membranes fuse together. This releases the viral capsid into the cytoplasm. Now, before more viral particles can be produced and before biosynthesis, HIV has to take its plus single-stranded RNA genome and convert it into DNA, and that's going to occur with the virally encoded reverse transcriptase, which is an RNA-dependent DNA polymerase. This DNA will then be integrated into the host genome by the viral encoded enzyme integrase. At this point, we call the virus a provirus. This particular provirus then can either undergo replication with the host's DNA polymerase or can be transcribed with the host's RNA polymerase. And after transcription, proteins from the virus can then be translated. There are a couple of important things to note here. First, HIV contains 2 copies of its single-stranded RNA genome. The second important feature is that reverse transcriptase makes a lot of errors. In fact, it is presumed that for every time plus single-stranded RNA is converted into DNA, reverse transcriptase will make 1 mistake in the reverse transcription process. This may actually explain why HIV carries 2 copies of its genome. For example, if one copy contains a mutation in the integrase enzyme, which renders it non-functional, then the other copy, which may contain a normal or functional copy of integrase, can still produce the protein without affecting the infection. In other words, HIV somewhat functions like a diploid virus. Once upon a time, being infected with HIV was a death sentence. But more recently, with the generation of numerous different types of antiretroviral drugs, we have found that people who have HIV can live just as long as individuals who do not have HIV, so long as they have access to medication and to good public health. There are a number of different targets for antiretroviral therapy, which is sometimes abbreviated ART or HART. For example, blocking the entry by blocking the interaction between the spike protein and the co-receptor CCR5 or CXCR4 may block the ability of the viral particles to enter into host cells. The earliest of antiretroviral drugs were reverse transcriptase inhibitors, which blocked the function of the viral reverse transcriptase enzyme. Other inhibitors may block integrase, the maturation of the virus, or the viral protease. The viral protease is an enzyme that cuts immature proteins into their mature form as the virus or those viral particles are exiting their host cell. Even though we have successful use of antiretroviral therapy, individuals who are infected with HIV will be infected with the virus for the remainder of their life. And that is because of HIV's ability to integrate its genome into the genome of our host cells or our T cells. What you're looking at here is a graph which compares the levels of CD4 lymphocytes or T helper cells in blue to the amount of HIV viral genome copies per milliliter of blood plasma. You'll notice that the infection of HIV follows 3 different stages. The first, which occurs from 0 to 12 weeks, is known as the acute stage. In this stage, we find a very rapid increase in the number of viral particles, as HIV is basically undergoing lytic infection, and we find a dramatic decrease in the number of T cells. After this period of time, the amount of HIV within the blood plasma will decrease, and HIV will have what we call a clinical latency phase, where not many viral particles are being produced. However, when they are, they will result in the death of T cells. And so you can see that over years, the number of T cells decreases while the number of viral particles that are detected increase. By year 8 in this particular graph, what you will see is that this individual has reached the 3rd phase of HIV infection, which is known as AIDS. or acquired immunodeficiency syndrome. AIDS is clinically defined by T lymphocyte numbers. When the number of T helper cells drops below 200 per microliter of blood, an individual is said to have AIDS. People who are infected with HIV and have AIDS do not die from HIV. Rather, their immune system is compromised because they have low numbers of T cells. These individuals often die from opportunistic infections, or infections that normally wouldn't do much other than cause maybe a mild illness in healthy individuals. Opportunistic diseases can include Kaposi sarcoma, which is caused by the virus Kaposi sarcoma herpes virus, Burkitt's lymphoma, which is caused by the same virus that causes mononucleosis or the kissing disease, and even bacterial or viral infections that could typically be treated. We might find individuals with AIDS could succumb to those diseases. Now, in individuals that are treated with antiretroviral drugs, we might find that the amount of viral genomes that are detected in an individual are at below detectable levels for many, many years, on the order of 20 to 30 years in some cases. However, because HIV has an error-prone reverse transcriptase, what we will find over time is that even individuals who are on antiretroviral therapy may need to change that therapy to protect themselves from the virus continuing to replicate within their body. Antiretroviral therapy is continuing to drive biomedical research as we search for new ways to treat and potentially to cure HIV.